Recombinant DNA technology is a method in which DNA molecule made in vitro with segments of two or by more different DNA molecules.
DNA is a sequence of DNA containing gene of interest. Gene of interest is the gene that we want. This is the gene of interest with the target DNA. This gene of interest we will cut using restriction enzyme, and this is the segment that will be produced. This gene of interest is very important for pest resistance, for growth, and for cleaning up toxic waste.
Enzyme which cut DNA molecules at a restriction site and produces short restriction fragments. Restriction site is the short base pair sequence recognized by a particular restriction enzyme. Restriction sites contain palindromics, where the same sequence of bases is found on both DNA strains but in opposite direction.
This is gene of interest which occur the sticky end. So the restriction enzyme cut the sugar phosphate backbone at each error. It started from G to G. So this is after it cuts the sugar phosphate backbone at each error. DNA fragment from another source is added. This pairing of sticky end produces various combinations. Fragment from different DNA molecule cut by the same restriction enzyme. So this is how the fragment from different DNA molecule come and attach to the gene of interest. So one possible combination occur. DNA ligase seal the strain. The recombinant DNA molecule is completely occur with sticky end. This is gene interest that occur in blunt end in the small one. So the restriction enzyme will cut into half at the pair. It will cut into half and divide it into two. A fragment from another source is added. The pairing of blunt end produces a various combination. Fragment from different DNA molecule cut by the same restriction enzyme. So the fragment from different DNA molecule will come attached to the gene of interest. So the DNA ligase seal the strain and the one possible combination occur. So the blunt end cut is happening.
Example of restriction enzyme. It divided into three parts. First, EcoR1. Second, small one. Third, BAM-H1. For EcoR1, it produced by E. coli. It recognized the base sequence of 5' GAA TC 3'. It also makes staggered cuts within the sequence. It produces sticky ends as a single-stranded ends. Second, small one. It produced by Serratia marcescens. It recognized the base sequence of 5' CCC GGG 3'. The cut is made between the adjacent C and G. Cut straight across the double helix. Produce blunt end. Third, BAM-H1. It produced by Bacillus amyloculifaxin. It recognized the base sequence of 5' GGA TCC 3'. It makes staggered cut, and also it produces a sticky end.
Plasmid acts as a cloning vector because it's able to carry foreign DNA and must have multiple cloning site MCS for a specific restriction site for cloning. Then it has able to replicate freely in the host cell and have selectable genetic marker as a resistant to antibiotic.
Next is, we have to know there are two types of modifying enzyme, which is DNA ligase and Taq polymerase. But in this case, after plasmid accept foreign DNA, DNA ligase will be used to join the foreign DNA fragment containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA.
Host cell is a cell which receives foreign DNA and allows the cloning vector of recombinant DNA. Host cell has three characteristics. to receive recombinant DNA through transformation process and able to amplify the gene product from the recombinant DNA. Able to maintain the structure of recombinant DNA from one generation to another generation. When the cell does the cell division, the daughter cell structure will be the same as the parent cell.
Besides that, we have to know that bacteria is the most commonly used as host cell because DNA can be easily isolated from or introduced into a bacterium. Bacteria culture grows quickly, rapidly replicating any foreign genes they carry. The example of bacteria is E. coli.
Gene of cloning consists of 5 steps. The first one is isolation of gene. Next, cleave and cut. After that, insertion. Then, transformation and amplification. Finally, step is screening.
The first step is isolation of gene. Plasmid from the bacterial cell is isolated, and the gene of interest is isolated from DNA of the donor cell.
Next is cleave and cut. DNA donor and plasmid bacterial is cleaved using compatible restriction enzyme, which is EcoRI. Make a single cut within the LEXZ genes, which is site of cleaving.
After that, insertion. DNA fragment of gene of interest is inserted into the plasmid, joined together by DNA ligase, forming recombinant DNA.
Then, transformation and amplification. The recombinant DNA then insert into the bacteria through transformation process. As the bacteria reproduce, the recombinant DNA is also replicate.
The final step of recombinant DNA is screening. The screening process is done by using blue-white screening technique to identify bacteria that carries the recombinant DNA.
The plague bacteria in agar containing antibacterial penicillin and agar. It divided into 2 types of antibiotic which is not resistant and resistant to the antibiotic ampicillin. Is the resistant to antibiotic ampicillin, the bacteria will reproduce and from colonies of the bacteria. So from here we can see if the colonies turn the color into blue, which means the color are without recombinant plasmid. Somehow the colonies with recombinant plasmid will be white color.
The reason why the colonies without recombinant plasmid change to blue because the plasmid contain intact LEXZ. It can produce beta-galactosidase and hydrolyze at skull. Why the colonies that change the color to white? Because the recombinant plasmid do not contain intact LEXZ. Also, it cannot produce beta-galactosidase and hydrolyze at skull.
So this is the plate where shown the final step in screening process. The white color has recombinant plasmid and the blue color don't have the recombinant plasmid.